Hi everyone. Thanks for Twist and the team. I'm here to present you about the Twist custom and cost-effective NGS panels experience from MedGenome. I'm Shaktivel, VP Lab Operations of MedGenome. This is the agenda, just a short agenda or introduction to MedGenome labs and requirements of an optimal NGS panel, and just describe what are our panels from Twist and the validation process and some two example validation results to really show how Twist has helped us in having some clinically validated tests. Coming about the MedGenome labs, so we were founded in 2013 as the first provider of NGS to the Indian market, and have been rapidly growing over the last 7 to 8 years, and now are the India's market leaders in genomics, far ahead of the competition both in scale and capabilities. We have been able to introduce a lot of tests, done more than 150,000 tests till date, more than 400 plus different tests, not only on NGS platform, but also on other platforms like immunohistochemistry, flow cytometry, microarray, etc. And we were the founders of Genome Asia 100K, an initiative to sequence 100K individuals of Asian descent, which was published recently. The first set of results were published recently. And we continue to innovate tests across portfolios and ensuring last mile delivery. These are the key milestones starting from 2013 to 2020. I'll just say a few of them. We started with exome testing sequencing in 2013 and along with some small panels on the MySeq and HiSeq 2500 platforms. And now we have grown into a CAP accredited test for more than 400 plus tests, which are performed under one roof. We have validated, published, and introduced several new tests to the Indian market, like the liquid biopsy test, the NAPT, non-invasive prenatal testing on NGS. We were the first ones to give HLA typing on NGS, the high resolution HLA typing on NGS. And very recently, we published the coronary artery polygenic risk score. We started as a $10 million company, and now we are a $300 million company. And just a few more points here about our achievement positions. And we have 1,600 plus hospitals collaborating with us, sending samples, providing access to samples and patient records. And we generate close to 1 petabyte of their data in diagnostics till date. And 100 bioinformaticians, that is our USP, 100 bioinformaticians with the software algorithm which are proprietary to our company. The depth of our experience and proprietary technologies has been very critical for our growth. As I said, we have more than 475 plus tests and different platforms, and we have a proprietary tuberculosis test for drug-resistant detection based on next-generation sequencing. Comprehensive test menu covering all disciplines of medicine. And we have 4 different business segments: the Claria, Actia, Prima, and Micra, catering to all different aspects of medicine. And this makes us easy to reach the clinicians, reach our patients, and ensure that almost all of them are benefited. Different genomic platforms, starting from Illumina MySeq to Illumina NovaSeq. We have the HiSeq X10, and also all platforms for microarray, including iScan and Fluidigm and Affymetrix. And as I told you, we have our own individual, custom pipelines for bioinformatics. So this gives us an ability to analyze gene panels, exomes, and whole human genomes for diagnostics and research, and combines algorithms with in-house curated literature content and clinical and disease knowledge databases. We are accredited by CAP, EFI. As per ICMR regulations, certain tests we are accredited by EMQ also. These are our different panels which we have used over the years, and our growth in terms of NGS panels. We started with only exomes, which was a sure select exome in 2013, and very small gene panels. analysis on Excel sheets, and now we have grown by 2021, where we have our own custom built softwares to do interpretation and reporting. So 2014, we also introduced the clinical exome to Indian market. We were the first to introduce the clinical exome to Indian market, and it was a huge hit. This was all on initially it was all on an Roche Nimulgen platform or chemistry. Several different panels depending on the requirements are also introduced, and over the years we have modified or we have revised the content of our clinical exomes to clinical exome V5, which is running now. And we were able to have hybrid capture panels for MTB, for carrier screening, and all this happened on the Nimulgen platform only initially. We also introduced comprehensive tumor panels initially on the off-the-shelf panels like TST170, but currently we have totally moved to custom panels which are based on Twist. From since 2020, we moved our exomes to Twist exomes, comprehensive exomes, clinical exomes, the V4 and V5 also on Twist platform, and all our comprehensive oncology panels on also Twist platform. Almost 90% of our NGS samples are processed using the Twist library preparation kits. And as per our recent stats, in the last 1 year, we have processed close to or more than 35,000 samples on the Twist panels. What are the requirements of an optimum NGS panel? So this is critical. Before we decide to do or optimize a test, we need to analyze and identify which kits to use. So there are two commonly used NGS panels: the hybrid capture panels and the amplicon-based panels. And these are the common companies or vendors who give the different panels. So the hybrid capture panels are mostly by Agilent, Twist, Illumina, and IDT, and the amplicon-based panels by QIAGEN, Thermo, Amoidx. Recently, there are some MAP-based panels also, molecular immersion probe-based panels. And these are the key differences between the hybrid capture and the amplicon-seq panels. So I would say the main difference is the size of the panel. The hybridization capture panel is virtually unlimited by panel size, whereas the amplicon sequencing panel, we cannot have more than 10,000 or even 20,000 amplicons in per assay. So that sort of limits the size of the panel or the size of the region which we are looking to target. And though it is more sensitive and more specific, I would say this limitation makes its usage very limited. So cost-wise also, there's not much difference between hybridization capture and amplicon sequencing panel. I would say hybrid capture panels are much cheaper and best suited for clinical applications which are which have to be cost effective. What are the characteristics of a panel for clinical use? I have put the cost effectiveness as the first point, mainly because we in India have to cater to a very cost sensitive market, and if we don't have cost effective kits, it's very difficult to sell the panels. It does not reach all our patients which we intend to reach, and more importantly, these tests are not covered by the insurance. So another thing is minimal multiplexing. This is very critical because as we multiplex more and more samples, depending on the quality of the samples, there is a chance that one sample or two samples within the pool doesn't give sufficient data, and this will result in reprep and resequencing and increasing cost. So I would prefer, as a customer, I would prefer to have minimal multiplexing, and most of our test panels are fourplexed. We have optimized for fourplexing, and there are a few somatic panels which we have optimized for twoplexing also. The good range, when I say good range, the probe size or the probe numbers have to be of a big range. So if you look at the hybrid capture panels, it can be anything from a few kbs to several mbs, and that has to be covered well by the panels. There should be short library preparation timings because these are all short tag tests. Patient wants the results as early as possible. So the time taken from DNA to data has to be as minimal as possible. And initially, the hybridization timings used to be 16 hours and 72 hours. Currently, now with the newer chemistry, Twist has brought it down to anything between 15 minutes to 4 hours. There should be a good coverage of the region which we are trying to target, and the on-target reads need to be high. This is very critical because the lower the on-target reads, the higher is the loss of data. Minimal loss of data due to other reasons like adapter dimers or duplicates should be as low as possible. We prefer tiling probes. There are also non-tiling probes which have one probe per exon. Tiling probes ensure that the coverage is very good, and even if one probe doesn't work, the other probe ensures the region is covered well. So we prefer tiling probes 1x for germline and 2x for all the somatic panels. Additional requirements of any panel will be FFPE compatible, considering the quality of DNA we get from FFPEs are not very good, especially so in countries like India where there is no proper processing of the tissue samples. We get very highly fragmented DNA. So the panels have to be compatible with FFPE. UMIs are becoming very important now in the somatic panels and the cfDNAs, wherein very low frequency variants also need to be picked up. So UMIs are very critical, and happy to know that Twist is coming up with UMIs now. And unique dual indexes increases our capability to multiplex many samples in a run, and have become a norm for most of the panels in the market now. What are our panels from Twist? So as I mentioned, MedGenome has most of the panels now on the Twist chemistry only, except for a few of the shelf kits which we use for specific assays like TMB, etc. So our germline panels include clinical exome, the updated clinical exome, which we call it the version 5. The comprehensive exome, which is an off-the-shelf kit from Twist, is our exome in use. And we have other panels like MODY, muscular dystrophy, hypercholesteremia, and specific panel for PGX also. The somatic panels include the comprehensive solid tumor panel and the liquid tumor panel. We have separate family panels for DNAs and RNA. The DNA panels for detecting SNPs and CNVs, and the RNA panels to detect the fusions. We have validated and completely validated and moved to production both the panels, are DNA and RNA panels of the solid and liquid tumors. We have also recently purchased a panel for liquid biopsy testing for both SNVs, CNVs, as well as fusions. It is yet to be validated. What is a typical validation process which is used across the globe, and which is used by us more because we are a CAP accredited lab? Whenever we get a panel in the facility, the first thing we do is the verification to look for the probe performance. We usually run the NA12878, which is a sample with the truth value and a few healthy controls. We see the assay performance in terms of the loss of data, in terms of coverage, in terms of individual gene coverages, the on-target reads. And once they pass all these criteria, then we take it for complete validation. And where we run for germline samples, minimum of 20 clinical samples for SNPs and for SNVs, and 20 clinical samples for CNVs. And for somatic panels, we run much more samples so that all the critical mutations are covered. And we need to check the sensitivity, specificity, repeatability, reproducibility, and accuracy with either by running our PT, CAP PT, or by doing a EQUAS. And CNV analysis pipelines have to be standardized before they are the test is sent to the market. So coming to some example validation results. So I've just selected 2 example validation results here to show how Twist panels are performing. So one is a off-the-shelf panel, and another one is a custom panel. The off-the-shelf panel which we have used and validated is the Twist comprehensive exome panel, which consists of both the core exome and the refs exome put together. So Twist had come up with the core exome panel and the add-on the refsic exome, which can be added on to the core exome to form the comprehensive exome. And this is the content of the panel, which can be got from the product specification sheets. The Twist core exome is 33 MB, and with the add-on it becomes 36.6 MB or around 37 MB. And these are all 120 base pair dsDNA probes. All twist probes are similar of this size only, and 90% of the probe sequence within the target region is being designed to minimize sequencing. So this can be got from these specification sheets, and this is additional details. Wherein we show that the twist comprehensive exome is a mix of both core exome and RefSeq panel, and the core exome is a part of the comprehensive exome. This covers almost 200 more than 200,000 genomic regions, mapping to 240,000 CDS regions, and almost 24,700 genes, 20,542 coding genes, and 4,156 non-coding genes. This is the panel validation workflow used by the bioinformatics team. They have the panel bed file and the CDS bed file, which is integrated and into the germline analysis pipeline. The raw FASTQ from different batches are run through this pipeline to arrive at a QC summary, concordance of the GAAB sample, variant concordance of the GAAB sample for both SNP and indels, and identified variant concordance for the patient samples or the clinical samples, IGV concordance for all the variants which have been detected, and coverage summary for all transcript CDS gene and primary transcript CDS genes. These are some of the quality parameters which we check. If you can see here, the total data generated, the adaptive data loss. So you can see there was a slightly high adaptive data loss. It's close to 16.5% here, or 16.5% here. The duplications is within limits. It's around 6%. The median insert size is around 230 to 250 base pairs. The total pass alignment and the read on target percentage were all good. If you can see the read on target percentage, it is around 80%, which is very good to ensure that the loss of data is quite low. And CDS on target percentage is also quite good. So we got overall the metrics were quite good, and except for the higher 80 loss of data, which we were able to see a change as we went ahead with our complete validation. This is the data generated versus panel average depth. So this is also done to see how much of data we will need to generate to attain 80 to 100x panel average depth. This is per base depth. This is very critical because within MedGenome, for diagnostic purpose, we target 80 to 100x average depth, and we saw that with the twist exome, anything between 7 to 8 GB data gave us a good more than 90x average depth. This is the data QC summary for the comprehensive exome, and we can see that the key advantage being at the depth of more than 20x, CDS coverage is consistently more than 95% across samples, and this is what makes twist panel stand apart from the other panels. And the CDS of all transcript panel length is almost 36 MB, and all these are covered very well. This is another summary where we see at a 99% cutoff of 20x depth, we see close to 80% of the genes covered well. And similarly at 30x, that is also around 78% is covered. So 79% is covered, which means that the average coverage across the panel is very good. We compared this with the previously used exome because these are all clinical diagnostic tests, and to move from one test to another, we need to ensure that we are not going down on quality. And to summarize, we saw a general performance was comparable with the AGV5, and the coverage of in AGV5, the NA12778 is much more than in the average of 10 samples. And at 95% cutoff, the total number of genes covered in AGV5 was less than the twist. So you can see that there's 18,814 genes covered in AGV5, but in a twist it was around 984. This even if the AGV5 gene content was much higher than the twist RefSeq gene panel. This is another statistics where we see the overall genes covered in twist is slightly more than that in AGV5. The coverage of genes present in OMIM, HGMD, ClinVar, and our clinical exome was slightly less in twist than in AG5, which means that the clinical clinically relevant genes were covered better in the AG5. However, more or less the coverage is comparable in both the panels, and so we decided to go ahead with using the twist core exome or comprehensive exome panel. This is another statistics which you can see that the more number of genes are better covered in twist RefSeq than in AGV5, as seen with compact lines towards the high coverage in the left plot for twist RefSeq. This is similar. I'll just skip through this. To summarize, twist exome panels were better than the SSV5 panel in terms of performance. However, SSV5 panel was slightly better in terms of overall content, which resulted in larger size panel, and hence larger amount of data has to be generated. Coming to the next validation, which was a fusion panel, which was custom panel, which was designed by MedGenome. And this was an RNA-seq panel targeting 2,377 targets in 160 genes and 10 upstream regions. The panel length was around 431 kilobase pair. We ran around, in our first batch of validation samples, we ran around 19 samples with known different fusions, and these were the QC plots for them. So most of the QC parameters passed, and they were all within the acceptable range. And this is based on the QC parameters. We were able to generate QC cutoff tables for different parameters, and pass, alert, and fail parameters were cutoffs were generated. And among the 19 samples, 16 samples passed the QC cutoff and 3 were in the alert range. There was no sample which failed the QC cutoff. And we saw that the fusions detected on the samples were concordant with the previously available results, which were tested on another platform, which is RT-PCR. And we were able to identify the fusions in all the 3 different softwares like Fusion Catcher, Fusion Map, and Star Fusion. All the 19 samples showed good concordance and good read depth. And the concordance was not only good in the batch 1, the other next 2 batches which performed also, it was good. We took the testing further, a little further, to see if 2 hours hybridization time was good or as good as 4 hours hybridization time, which was used for our first batch of samples. So to test this, we ran 4 samples at 2 hours and at 4 hours hybridization time, and we saw that generally the quality was good, data quality was good for both, and the concordance was perfect. So on using all the 3 tools, the concordance of the 4 fusions in the 4 samples was there between the 2 hours and 4 hours. We concluded that though the concordance was good for fusion events, 4-hour samples were observed with better read support than 2-hour samples. But considering the TAT, for most of the leukemia samples, we need a lower TAT. We finalized there with 2 hours protocol. So to summarize, the performance of the twist panels were good for clinical use for both germline and somatic panels, DNA as well as RNA panels. It was cost effective compared to most suppliers in the market, have several multiplexing options, good for fusion detections also, and also CNV detection. There was even coverage across the targeted region, and support and supply were good. These are the acknowledgements. I would like to acknowledge my team and the twist team. Thank you.